Good morning. Good morning, everyone. Welcome to the Clinical Laboratory Fee Schedule Annual Laboratory Meeting. My name is Rashida Arthur, and as a member of the Medicare Clinical Laboratory Fee Schedule Policy Team and the Division of Ambulatory Services, I will be facilitating today's meeting. I would like to begin by expressing appreciation for those who are able to attend today's meeting in person and virtually. I'd also like to extend a warm welcome to members of the Medicare Advisory Panel on Clinical Diagnostic Laboratory Tests who are also on the line and may participate by posing questions to presenters throughout today's meeting. Here you can see our agenda for the day. As always, we have a 10-minute time limit for each presentation, followed by any questions posed by the public or the panel. However, we understand that presentations may not last the entire 10 minutes, therefore the meeting may move along quickly at times. We'll get through as much as we can during the morning session before lunch. Therefore, we recommend that even if your lab or group is slotted towards the end of the agenda, keep an eye out for the progression of the meeting, just so you can be prepared for when it's your turn to speak. The CLFS annual public meeting provides an opportunity for the public to present and comment on the appropriate basis for establishing payment amounts for new or substantially revised healthcare common procedure coding system codes being considered for Medicare payment under the CLFS for the next calendar year. This meeting also provides a forum for the public to provide comment on certain reconsideration requests submitted during the previous year regarding final determinations on new and substantially revised test codes. Please note that we will only be discussing the codes addressed in our CLFS CY 2027 test code updates file, which is found on our CLFS annual laboratory meeting webpage. Comments and recommendations on whether codes identified as gap fill codes from last year's final determination is a separate and distinct process that will not be discussed at this meeting. That said, MAC preliminary gap fill rates are currently up online on our public meeting webpage. We will be accepting public comments on those preliminary MAC recommendations until July 1st, 2026. I'd like to share some logistical information regarding the flow and format of today's meeting. Okay. As you may notice, we have a packed agenda with approximately 47 speakers from various specialty societies and laboratories. We thank you in advance for your prompt delivery of your presentation in the 10-minute time frame. Given the number of presentations, we'll be pretty strict with not allowing presentations to exceed the 10-minute mark. As I mentioned earlier, we can't predict the exact time your group will be up, so please keep an eye out on the progress of the meeting throughout the day so you'll be ready to speak. After today's meeting, a recording will be posted on the public meeting website on cms.gov. At the conclusion of each presentation, we'll have a very brief, maximum of 5-minute open forum for questions to presenters. We will facilitate this process using the Q&A function located on the Zoom portal, or if you're in the room, simply raise your hand and we'll get a mic to you. We have staff members that are monitoring the Q&A box, so please write your question there so we may read it out loud. If we see no comments, we'll simply proceed to the next speaker. Again, just please keep your remarks brief and restrict any comments or questions toward the presentation on the new codes for 2027. We will not discuss any policy during this meeting. Consistent with the annual CLFS process, after this public meeting and with the assistance and guidance of the CDLT panel, we will determine the basis of payment, either crosswalk or gap fill, for each new or reconsidered laboratory test. Please refer to the CLFS website for more information on the upcoming CDLT FACA panel meeting, which is scheduled for July 14th and 15th. This year, we will host a second public meeting and a CDLT FACA panel meeting scheduled for September 15th and 16th. During the meeting, the public will have the opportunity to present recommendations on the appropriate basis for establishing payment amounts for CDLTs for which CMS received no applicable information during the data reporting period between May 1st, 2026 and July 31st in order to calculate Medicare payment rates. After the public provides its input, the advisory panel on CDLTs will provide its recommendations to the secretary and the administrator on payment recommendations for those CDLTs. After the consideration of the information from the ALM, the public comments, public meetings, and CDLT panel recommendations, we will post preliminary payment determinations on our website in late or early October. We will accept public comments for 30 days following the posting of those preliminary payment determinations. Unlike a new crosswalk test, the payment amounts for gap fill test is not established when we determine the basis for payment because it takes about 9 months for CLFS annual public meeting for MACs to establish MAC specific amounts and report those to CMS. In November of each year, we generally finalize the basis of payment for new and substantially revised test codes and the amount of payment throughout the annual CMS instructions, implementing the updated CLFS for the next calendar year. The public has additional 60 days from the date we issue our final payment determinations to request reconsideration of either the basis of payment or the amount of payment for new and substantially revised test codes. The public may comment on those reconsideration requests at the next CLFS annual public meeting in June of 2027. Once a code or a lab test has undergone a reconsideration cycle and CMS has made second final determination on the code or test, the payment methodology is final. For any new tests that will be gap filled, we ask our Medicare contractors to develop a carrier-specific gap fill amount by April 30th of the following year. We aim to finalize by September 30th of that year. And unlike tests crosswalk to a CLFS lab test, the payment amount for a gap fill test is not established when we determine the basis for payment because it takes about 9 months for the contractor to establish carrier-specific amounts. After pricing for gap fill codes are developed by the Medicare contractors, we'll post on our website and establish comments for about 60 days. Later in the year, when the gap fill amounts are posted on the website as final, we accept reconsideration requests on the gap fill amounts for 30 days. And once the reconsideration process is complete for a cycle, the determination is final and will not be subject to further reconsideration. Okay. Last, I'd like to share a few housekeeping items. Please note the following. The agenda, code list, and presentations may be found on our CMS webpage, previously noted. If you are a speaker, please speak directly into your microphone, announcing your name before speaking. And virtual presenters, please place your phone on mute when you are not speaking. For any comments or questions, please walk up to the microphone or type the question in the Q&A chat function. The questions will be posed after each presentation. For each presentation, there's a 5-minute limit for comments or questions. If you have any questions during this meeting, please do not hesitate to reach out to a member of the Division of Ambulatory Services team. If you have questions following this meeting, please submit inquiries to the annual laboratory meeting resource box or CLFS_annual_public_meeting@cms.hhs.gov. Before we begin with the presentations, I'd like to introduce the management team of the Division of Ambulatory Services. Sarah Shirey Loso, the director. Michelle Cruz, deputy director. And finally, Mathali Thayal, the acting deputy director of the Hospital and Ambulatory Policy Group. This concludes my introductory and housekeeping comments. Now we'll hear a few remarks from Mathali Thayal. Following her remarks, we'll begin with our first presenter.
Good morning, everyone. I'm Mathali Thayal, and as Acting Deputy Director of the Hospital and Ambulatory Policy Group, I want to welcome you and thank you for participating in this year's annual meeting on payment methods for new and reconsidered clinical diagnostic laboratory test codes for the calendar year 2027 Medicare Clinical Laboratory Fee Schedule. As always, CMS sincerely appreciates the public and the panel's expertise and values its contributions to improve CMS policy by strengthening the connection between Medicare payments and high-quality, efficient care for our beneficiaries. Section 1834 AF3 of the Social Security Act requires CMS to hold a public meeting to receive comments and recommendations from the public regarding the basis for establishing payment amounts for new or substantially revised laboratory test codes. CMS has held public meetings annually for the CLFS since 2005. As a reminder, on February 3rd, 2026, Section 6226 of the Consolidated Appropriations Act of 2026 was passed, which updated the data reporting requirements for CDLTs that are not advanced diagnostic laboratory tests. It also delayed the phase-in of payment reductions under the CLFS from private payer rate implementation. Beginning January 1st, 2027 through 2029, payment may not be reduced by more than 15% per year compared to the payment amount established for a test the preceding year. We are currently in the data reporting period, which ends on July 31st, 2026. The data reporting system has been open since May 1st and is accepting data. Please remember to report data based on the updated data collection period of January 1st, 2025 through June 30th, 2025. There are many resources on our website to assist the labs in reporting, and please reach out to any of the CMS staff here today or visit our website. We are very excited to note that we are already receiving data and are looking forward to more. We have approved 18 ADLTs. Another round of applications can be submitted through July 31st, 2026. And as always, we look forward to receiving additional applications. Finally, we are aware there are a few CPT codes that were recently established but are not reflected on today's agenda for presentation. We have received some incoming letters and questions on these codes. We are reviewing these concerns and we hope to have additional information soon. But for today, the agenda is set and those codes will not be discussed. So to the public and the panel, again, we sincerely appreciate your invaluable contributions of professional expertise and experience to this public process behind the clinical laboratory fee schedule. Input from you is an important contribution to the policymaking process, and we value your feedback. So looking forward to another productive meeting. Thank you.
Okay, first up, McDermott.
Good morning. My name is Deborah Goddess. I'm from McDermott Plus, and I'm speaking on behalf of Abbott today. I'm going to be talking about PLA code X300U. So X300U, as I mentioned, is for the Alinity Eye TBI panel from Abbott Laboratories. The descriptor is as follows: neurology, traumatic brain injury, analysis of glial fibrillary acidic protein, and ubiquitin carboxyl terminal hydrolase L1 or UCHL1. It's an immunoassay in serum or plasma. Individual components reported with the overall result of positive or negative based on the threshold comparison. In terms of the background about the test is a panel of in vitro diagnostic immunoassays for the quantitative measurement of GFAP and UCHL1 in serum or plasma, and then a semi-quantitative interpretation of the results derived specifically from these measurements. The immunoassay is intended to aid in the evaluation of patients 18 years and older presenting with suspected mild traumatic brain injury, with the interpretation of the test results in conjunction with other clinical information to assist in determining the need for a head CT scan. In terms of the resources required for this test, this panel is run on serum or plasma. It requires optimized FDA-cleared reagent kits, plus collaborators and controls. It is run on a dedicated instrument, the Alinity I. It returns quantitative results individually for GFAB and UCHL1, plus a semi-quantitative result, i.e. positive or negative, which is based on results relative to the cutoff measurements that have been set. In terms of our request, the crosswalk recommendation is to PLA code 570570U, which is for neurology, traumatic brain injury, analysis of GFAB and UCHL1 immunoassay, whole blood or plasma, individual components reported with overall results of elevated or non-elevated based on the threshold comparison. The rationale for this crosswalk recommendation is that there are similar analytical characteristics. It's the same analyte type of protein. Both are immunoassays. It's the same number of targets and actually the same targets. It has the same post-analytic characteristics with separate reports individually of the quantitative reporting of the individual analytes, followed by the semi-quantitative characterization of the results based on interpretation of the results. And it is in the same clinical space. As the table shows, these tests are very similar. They're nearly the same test, just simply on a different platform. Both are on whole blood plasma or serum. They're both the same methodology, both same target type of protein, same number of targets, both with individual quantitative results, followed by a semi-quantitative result based on interpretation of those. And for those reasons, we are requesting crosswalk to 0570U. With that, I'm happy to answer any questions. Thank you.
I'm here to discuss PLA code 0648U. This test is manufactured and distributed by Thermo Fisher Scientific. Good morning. My name is Rob DeMonaway. I'm Director of Reimbursement Strategy with Thermo Fisher Clinical Sequencing Division. And being this early into this morning's agenda, we hope to keep this moving along quickly and keep us on track. This session is focused on PLA 0648U, the Oncomine DX Express test. The code descriptor reads oncology, solid tumor, targeted genomic sequencing analysis to detect deletions, insertions, and substitutions in 42 genes, copy number alterations or amplifications in 10 genes, and fusions and splice variants in 18 driver genes from DNA and RNA extracted from formalin-fixed paraffin-embedded tissue. This test received FDA approval and Medicare coverage via NCD 90.2 on July 2nd of last year. It secured a DEX Z-code identifier on November 11th of last year, and its PLA code is effective July 1st of 2026. I'd like to underscore a few key facts about this in vitro diagnostic test, which uses NGS technology to inform timely oncology therapy decisions from DNA and RNA. The FDA approval included both a tumor profiling claim and companion diagnostic claim for EGFR exon 20 insertion, mutation positive non-small cell lung cancer patients. And 0648U operates on the Genexus DX sequencer using ion torrent technology. Now is when you typically hear about the slow, labor-intensive, complex requirements of the steps required to perform or to sequence FFPE tissue. What's similar this morning? Those steps that are required: library preparation, sequencing, bioinformatic analysis, and reporting. What's different? The Genexus DX system introduces automation and efficiency at all stages, and we'll explain the impact of that automation on the next slide. The benefits are significant: a reduction in hands-on time for lab technicians, an improvement in reproducibility. Sequencing results are possible in 24 hours. And with the sample input requirements of 10 nanograms of DNA and RNA, 20 total, Oncomine DX Express test maximizes success with a small and poorly characterized tissue samples which present frequently in beneficiaries with advanced cancer diagnoses. Our recommendation 40648U is a crosswalk to PLA code 0022U, the Oncomine DX target test. Similarities include both use ion torrent technology to sequence DNA and RNA via FFPE samples. Regulatory status, both are FDA-approved tests with companion diagnostic claims, and both are distributed IVD kits. This table may be a bit of an eye test, but it explains our crosswalk rationale in greater detail using 0022U, 0523U, and 0648U for comparison's sake. What's common is that each assay is FDA-approved, distributed NGS testing with a companion diagnostic claim. Notable differences may be seen in a few key areas, though. Outliers are shown in red. The number of genes reported. 0022U reports 23, 0523U reports 22, and 0648U reports 46. Second, tumor profiling claim. 0022U does not have one and is limited to non-small cell lung cancer indications. But 0523U and 0648U do and are nationally covered for several solid tumor indications. Last is RNA biomarkers and fusions. 0022U and 0648U include them. 0523U does not and was priced accordingly last summer. In closing, Oncomine DX Express test introduces expanded molecular content versus the two distributed IVD assays previously noted. It secured national coverage under Medicare's NCD 90.2 and generates rapid results through the Ion Torrent Genexus DX system. Thanks for the opportunity to present this morning, and I will take questions after my colleague has completed the balance of the Thermo Fisher segment.
Thank you very much. Could we have the next slide, please? Thank you. Good morning, everyone, and thank you for the opportunity to present our test today. My name is Jamie Ashby, and I am the Exent R&D lead in the Specialty Diagnostics Group at Thermo Fisher Scientific. Today, I would like to discuss PLA code X269U, which is the Exent immunoglobulin isotypes assay. Next slide, please. This test is an immunoglobulin isotype assay that combines an immunoprecipitation assay, MALDI-TOF mass spectrometry, and immunoturbidimetry to profile M proteins for disease severity and treatment response assessment. This is in patients with monoclonal gammopathies. The Exent assay is intended for the use as an aid in the evaluation of monoclonal gammopathy of undetermined significance, and as an aid in the diagnosis of smoldering multiple myeloma, and Waldenstrom's macroglobulinemia, and in addition, AL amyloidosis. The assay involves immunopurification, MALDI-TOF mass spectrometry, immunoturbidimetry, and proprietary software. Next slide, please. The Exent assay consists of first preparing and mixing serum sample and relevant reagents on a roller mixer, and then placing them on the Exent IP500, which is an automated liquid handler, for the immunopurification and moldy plate spotting. The sample is then distributed into 5 independent reactions to immunopurify each isotype using a proprietary anti-human sheep antibody, which is covalently linked to paramagnetic beads. The purified immunoglobulins are then eluted off the beads and reduced into their heavy and light chains, and then spotted onto moldy target plates. The plates are manually transferred to the Exent IX 500, which is a MALDI-TOF mass spectrometer where the data is acquired. Proprietary software is used to then identify the monoclonal immunoglobulins and to flag peaks from therapeutic monoclonal antibodies based on their known mass-to-charge ratio. The software also performs semi-quantitative analysis of the monoclonal immunoglobulins based on the peak area ratios from the mass spectra and the concentrations obtained by immunoturbidimetry. A final report is then generated by the software with the results. Next slide, please. Thank you. We are requesting that our test be gap-filled because there is no comparable code that our test can crosswalk to. The closest analog is code 0077U, but there are significant differences in these tests, which we believe makes a crosswalk inappropriate. Namely, X269U incorporates turbidimetry while 0077U does not. X269U provides semi-quantitative results while 0077U is a qualitative test, and X269U uses proprietary software while 0077U requires intervention by a laboratory professional at multiple points in the workflow. And with that, I'd like to thank the committee for their time, and I would also welcome any questions. Thank you.
At this time, we'll take questions. It appears we have some questions online. Dr. Bridges, do you want to go ahead and unmute your line and
Yes.
ask your questions?
Hi, I'm Dr. Bridges. I'm one of the CMS panelists and wasn't able to put the questions in the Q&A, but I do have some questions for the first Thermo Fisher test.
Sure, go ahead.
Yep. And so my first question is to if you could provide the full list of genes and by tumor type, which guideline recommended biomarkers are and are not covered for your test.
Sure, would be happy to share that offline through email, but would also quickly note that the NCD issued by Medicare covers this assay currently for 626 unique ICD-10 codes, but we'll certainly follow up with gene list and the requested information shortly.
No, I thought that your crosswork was reasonable. The other concern that I had was that I did not see that the tumor mutation burden and microsatellite instability was reported with this test. So I wondered how the immunotherapy relevant biomarkers would be addressed for the build indications without that data.
Yeah, so your observation is correct. This assay does not include MSI or TMB calculations.
And then beyond the EGFR exon 20 CDX claim, what additional companion diagnostic indications are approved or planned, and what tumor profiling claims are on label versus research use?
Super. We'll appreciate that question, and we'll add it to the follow-up items that we
Okay.
respond to there.
Sure. And what is the RNA fusion sensitivity from the formalin-fixed paraffin-embedded tissue, including minimum DV200 and the sample failure rate on the Genexus workflow?
Sure. It's also something I'm happy to follow up with, and information that's readily at hand. Thank you.
Right. And the last question that I have is how is it positioned against the established comprehensive panels on cost, tissue requirement, and turnaround time? And what is the concordance with those panels? Because it's a great panel, but it's fewer genes in this panel compared to the more comprehensive tests.
Sure. Okay. Well, we'll also add that one to the list. As you might guess, I'll more than overstep my time allotted, and don't want to finish this morning in rough shape with the Sarahs.
Okay, wonderful. Thank you so much.
Thank you. Next up, Barrett Diagnostics. Make sure your phone is unmuted or you're unmuted.
muted still. So, thank you. My name is Joseph Chow, Chief Medical Officer for Virant Diagnostics. We are presenting two codes. The first one is PLA 0604U. This is a bradykinin assay, total quantitative by liquid chromatography and tandem mass spectrometry. Next slide, please. The background of this test and why it's important is that it's basically a diagnostic test that is used in treating and following patients with a rare disease or orphan disease called hereditary angioedema. It affects about 1 in 30 to 50,000 people worldwide, which translates to about 6,000 to 10,000 people in the United States. It's an autosomal dominant genetic trait causing dysfunctional or decrease in C1 esterase inhibitor, which is a negative controller of various cascades in the body relating to inflammation, coagulation, and blood vessel integrity leakage. It's also in one case of the cascade, it increases the kinin kallikrein system cascade if it's not in high enough quantity, causing a rise in bradykinin. And bradykinin itself is a 9 amino acid peptide that actually has an effect causing the blood vessel release of fluids to surrounding tissue. It's helpful in tissue injury or myocardial infarction, ischemia, but in aspects where you don't have control of it due to the mutation seen in HAE, a elevated BK could be very lethal and has caused death in many individuals with hereditary angioedema in the past due to laryngeal asphyxiation and swelling of the mucosal surfaces. So I think that with that as well, there's also multiple treatments, both the acute form of the angioedema caused by dysfunctional C1 esterase inhibitor, and there's also prophylactic treatments available for patients that are diagnosed appropriately. So our test actually is measuring the key element of hereditary angioedema, which is the bradykinin, and patients with symptoms of angioedema, etc., that could be actually mistaken to be anaphylaxis, which is much more common, and to use that in a differential for hereditary angioedema or angioedema. This methodology is quite complicated because it uses a technique that has been used in the past, kind of putting together various published techniques of isolating bradykinin. And what it doesn't do is, what the other tests don't do that was in the past available is measure bradykinin accurately, because bradykinin itself has a very short half-life, about 30 seconds or less. And you can imagine that would be very difficult. And the tests in the past have actually done it by immunoassay. Again, very inaccurate. So there's really currently no commercial availability to measure bradykinin commercially, especially not by this technique. So this is a fairly proprietary technique using various techniques that's been published in the past. We came up with and combined the methodology into what you see here. The workflow is quite complicated because you have to isolate the proper bradykinin metabolites to begin with, and it goes through various processes, including cold activation and resuspension of the solid to get solid phase extraction prior to the liquid chromatography and mass spectrometry. And of course, then you have to deal with the interpretation. These are very specific isotypes that could be detected through various metabolites of bradykinin, but we actually have found that 4 are what we use in the quantitative quantitation of our total bradykinin. Next slide, please. So what we have found is that the carrier pricing, as reported by our regional MAC, Novitas, is $24.11, which is using a CPT code of 83789, which is a non-specific mass spec technique, usually served for like drug confirmation testing, which is fairly direct, with very little isolation of the components itself, and just really running the mass spec and looking at the peaks. And we've done that in our lab as well. And however, this is just much more complex. It's usually run one at a time. It takes anywhere from 16, 18 hours, run in duplicates as well, over 1 to 3 days because of the co-activation property. So we take a 1 to 3 day measurement and take on the average of those two measurements to actually come up with a more accurate overall quantitation of bradykinin. So what we're asking for is that since there really is no equivalent gap fill, equivalency to FICO, we decide to go with the gap fill and the cost justifications, which was sent in to our regional MAC. And I could follow up with a more detailed submission as well. Dossier is just, as you can see, the cost justification, all the various steps involved is quite extensive. So we're asking for a reimbursement for gap fill of $370 a test. I'll stop here for any questions on that, and we'll take that afterwards, I guess. So I'll present this. Sorry about that. I'll present the second PLA code, which is 0605U. Next slide. Yes, should be different slide. Didn't I just present this? We'll go to 0605U. Yes, this is it. So 0605U is TPSAB1 copy number variation, a diagnostic test using digital PCR for hereditary alpha-tryptopenia. Hereditary alpha-tryptopenia is also a autosomal dominant genetic trait found in about 4 to 6% of the general US population. The mutation causes an increase in copy number variation for the alpha subunit of the TPSAB1 gene. It usually causes elevated serum basal serum tryptase, usually above 11 or so, but in many instances, it actually would be normal by most standard laboratory reference interval, which is 8 or so, and that would be normal. So patients generally present with multi-system symptoms or constellation of different symptoms and even signs, including anaphylaxis, allergies, even GI symptoms, inflammatory bowel-like type symptoms, and other symptoms such as neurological related symptoms. So the gene itself is fairly hard to precisely detect due to its conservation. So you have to use a very sensitive technique. It's quite involved in setup using a digital PCR technique, which we have been doing for the past 2.5, 3 years or so. This is a laboratory developed test, obviously. And so what we are using is seen on the next slide. So obviously trying to get this from blood, whole blood processing, as you see the workflow using a digital PCR is also quite involved and takes approximately 5 to 7 hours of runtime, both instrument and labor costs, as well as specimen preparation, which includes specimen preparation. Then you have to have the result interpretation, which does total take about 2 to 3 days turnaround time. Again, being a somewhat rare disease, we're not getting a large quantity of orders from around the United States in our lab. And so I think this is usually run in tiny batches, if at all. Sometimes we have to run it one at a time, depending on how these actually come into our lab. Next slide. So we're recommending a gap fill because 81479, which has been previously used, has a variable reimbursement rate from 0 to as much as $1,000, $1,200 or so. So I think the variable cost reimbursement is something that we want to be very straight about, which is essentially that this test costs to run in our lab approximately $244.57. Again, this price justification and spreadsheets were sent to our regional MAC in Novitas. And I believe the carrier price for this was approved last year for the January point, but I think what was missing was my request for the shipment and costs and overhead costs. That was, so that 244 was just the actual assay cost for us. And so we would like to actually request a increase to $320 per test, and to see the work and cost justification, which I could resubmit to our MAC after this call. So because there's no specific, well, there is but it's a non-specific 81479 code. And considering that there is the ICD-10 code for hereditary apoptosemia, which is a form of mastocytosis or mast cell activation disease, there's an ICD-10 code of D89.44. And I think this test is actually important to distinguish and to get the most sensitive test which we provide.
I'm sorry, we are at time.
Questions. Yeah, and are there any questions?
Hi,
Thank you.
This is Chris Chong from the panel. So for 0604U, you're getting the results for 4 bradykinin peptides in one run, right? You're not running it 4 times.
That's correct.
Okay, thank you.
No more questions. I thank you, and I'll pass along to the next speaker.
Thank you.
Good morning, I'm Federico Monzon. I'm here presenting on behalf of CNSide Diagnostics on code 064U. Code 064U, it's a test for leptomeningeal metastases, tumor cell selection, identification, detection, and enumeration based on differential expression of multiple antibody markers. I'm not going to read them all. You have them on the screen. It's done on cerebrospinal fluid and is reported as detection and quantification of tumor cells. As a background, leptomeningeal metastases are seeding of the CSF or the leptomeninges from primary solid tumors, and it's a serious and often fatal complication of solid tumors. Unfortunately, cytology, which is the current method used to diagnose, has low sensitivity. It is very specific. So when it is positive, it's clear there is leptomeningeal metastasis, but it has low sensitivity. So the CNSide CSF tumor cell enumeration test was developed to more accurately identify patients who have leptomeningeal metastasis, enumerate and capture these tumor cells in CSF, allowing for earlier characterization of the suspected leptomeningeal metastases. This allows physicians to detect leptomeningeal metastases much earlier than what you can do with either cytology or imaging, and it allows them to adjust their treatment plans because a positive test is considered a positive leptomeningeal metastatic disease, and it directs treatment, and it directs also change in treatments and adjusting how the patient is treated. So the purpose I already mentioned is detection, characterization, and quantification of tumor cells in CSF of patients with suspected carcinoma or melanoma leptomeningeal metastases. The methodology is a proprietary collection tube, which uses a preservative shipped to a CLIA high complexity laboratory where the test is performed and analyzed. The tumor cells are captured on a proprietary single-use microfluidic device, and this is important. The microfluidic device is a single chip per patient per test, and it's proprietary. It's manufactured individually, uses a 10 antibody cocktail, which allows for the increased sensitivity, and then digital scanned, evaluated through automated finding of the cells, and then review by licensed personnel who are evaluating each and every cell individually to determine whether they are tumor cells or not. Oh, sorry. As far as resources, I already touched a little bit on these. It's a proprietary collection tube, individual microfluidic device that is used in a single use for each patient, cocktail of 10 antibodies to isolate the cells, isolate and trap them, a CLIA high complexity laboratory with a digital scanner, personnel that reviews and qualifies each individual run and each individual patient, as well as all that entails in a clinical diagnostic laboratory. We are recommending crosswalk to code 0338U. This is another code for identifying and enumerating circulating tumor cells by selecting, identifying, and morphologic characterization using a cocktail of antibodies, as well as what the CNSide test does, and uses quantification of HER2 protein biomarker expressing cells. It's done in peripheral blood. The rationale is that this code 0338U is the most comparable test existing in the CLF code. Both tests describe oncology use. Both tests describe circulating tumor cell selection in liquid specimens or body fluids, and both include detection, capture, and enumeration of tumor cells in these body fluids. Sorry. Both tests are comparable in terms of the methodology, which include immunologic tumor cell identification and enumeration, and also involves comparable resource utilization for capture reagents, digital imaging, and professional level review of both tests. As shown in this table, the resources and code descriptors are extremely similar. Both tests are done in a single laboratory. Both tests use a cocktail of antibodies that are used to identify and capture the cells. They are both done in liquid biopsy specimens. They both use proprietary collection tubes, very similar specimen preparation and methodology, using a selection, detection, identification, and morphological characterization, as well as both include algorithms to identify those cells and then report with detection and quantification of tumor cells. I will end with just indicating that CMS has consistently priced these tumor cell enumeration codes since the original code 0338U and 0337U. They were both priced as gap fill, and subsequent tests that have come to the market that have are very similar, capture and enumeration cells in liquid specimens, have all been priced consistently with crosswalk to either 0338U or 0337U. And with that, I will stop here and take any questions.
Hi, this is Chris Chong from the panel. Is there digital imaging and professional review for your tests?
I'm sorry, I couldn't hear about the imaging. Can you repeat the question?
Does your test require digital imaging and professional review?
It requires a review of a technologist, a PhD level scientist that does review each and every image that is captured from the cell. So it does require, it goes through a imaging that looks at all the cells, in a three-dimensional space, identifies the cells, captures those images, and then presents them for review for the doctoral level scientists to identify which ones are tumor cells and which ones are not.
Thank you.
Does that answer your question?
Yes, it does. Thanks.
Federico, it's Megan. Can you hear me okay?
Yes.
Quick question. Could you confirm, are there any additional biomarker labeling of these cells post enumeration, or is this just enumeration only?
This is just enumeration only. There is no additional subsequent biomarker identification.
Thank you so much.
I will point that while some of the other tests that have been crosswalk have additional biomarker identification, others do not. So it's a mixed group. Okay. Thank you.
Good morning, and thank you for the opportunity to speak with you today. I'm William Audie, the Chief Medical Officer for Agendia, and I'm here to discuss code 0630U. The descriptor is an oncology test used in breast cancer based upon mRNA gene expression profiling by microarray of 80 genes, 80 content genes, 465 housekeeping genes, utilizing formalin-fixed paraffin-embedded tissue, as well as an algorithm which reports out as an index, a diagnostic of a molecular subtype, either luminal, basal, or HER2 type breast cancer. I'll be referring to this test as Blueprint. The background of this test is that it was developed, optimized, and validated to provide clinically useful information regarding tumor behavior as well as response to therapy by classifying tumors based upon gene expression profiling into three distinct subtypes. Luminal type, tumors driven by the estrogen and progesterone receptor pathway, which may benefit from hormonal therapy. HER2 type breast cancer driven by the HER2 pathway, which may benefit, of course, from HER2 targeted therapies. And then a third type, basal type, which is not driven by either of these pathways or expressing receptors, therefore very similar to triple negative breast cancer with more aggressive biology and different responsiveness to therapy. The methodology of this assay is that it is a qualitative non-automated gene expression profiling assay, which evaluates, as I mentioned, 80 content genes by microarray technology using the total RNA isolated and enriched from an FFPE tissue section. It is indicated for use in so-called early stage breast cancer, stage 1, 2, or operable stage 3, node negative, or 1 to 3 positive lymph nodes, and tumors 5 centimeters or less in size. These results are then used by physicians in addition to clinical findings to inform treatment decisions, specifically systemic therapy decisions with targeted therapies, as well as timing of therapy relative to surgery. And my apologies for not having advanced the slide. Here was the purpose and methodology, which I just reviewed. Resources. So the resources used here, as I mentioned, begin with FFPE tissue samples being received in our laboratory, manually evaluated to ensure a minimum percentage of tumor cells that will yield sufficient RNA for isolation. The total RNA is then isolated, reverse transcribed into ctDNA, amplified and labeled. The amplified material is then hybridized into a DNA microarray and then analyzed according to a proprietary algorithm which allows us to determine the molecular subtype. These results are returned within 7 to 10 days to the clinicians in order to determine therapeutic strategy based upon this subtype. Our recommendation is to crosswalk to code 81540. This refers to an assay very similar to what I have just described, an mRNA-based assay where expression is determined by real-time PCR of 92 genes, 87 of which are content genes, and 5 of which are housekeeping genes, which is also used to classify tumors of unknown primary into organ of origin and subtype. Again, a subtyping assay similar to the assay I've just been describing. The similarities are shown here. It is a cancer subtyping assay, as is our assay. It is based upon FFPE with enrichment of RNA for the assay. It is a gene expression profiling assay, and it has a similar gene content. So with that, I'll stop, and I'm happy to answer any questions.
Hi, this is Mark Rumpler from the panel. So 0630U has approximately 465 housekeeping genes. Does that amount require any meaningful computational burden or analyst review associated with that compared to the crosswalk?
That's something I'll be happy to answer in follow-up. The large number of housekeeping genes has been standard for some time, but I'll be happy to follow up with an answer to that question.
Thank you.
Thank you. Thank you.
Okay, thank you.
Precision medicine care.
Good morning. Thank you so much for the opportunity to present. My name is Sherry Dadgar. I am board certified molecular geneticist, and I am serving as a laboratory technical director, along with Dr. Ehsan Nobach, who's a board certified nephrologist. We're presenting the PLA code C0628U for RenaDX product, which is a comprehensive renal disease genetic panel. To provide background, chronic kidney disease, also known as CKD, affects approximately 37 million Americans. Up to 20% of the CKD cases may have an underlying genetic cause. Major nephrology guidelines, including KDIGO, and recommend consideration of genetic testing in evaluation of chronic kidney disease. RenaDX is a comprehensive genetic test panel for CKD that utilizes sliced exome sequencing to identify diagnostic variants and risk alleles. It analyzes 449 clinically relevant and nephrology-related genes. Clinical impact of this panel includes accurate and definite molecular genetic diagnosis, changes or refinement of the diagnosis in a significant proportion of patients. It alters clinical management. Examples include avoiding unnecessary immune suppression, guiding targeted therapies, or adjusting plan planning.
I'm sorry, Sherry.
Yes.
It looks like there's a slight typo on the presentation. Could we hold off on your presentation and loop you back around after we figure out what's going on?
Sure. No problem.
Okay, thank you.
Of course.
Lumino DX.
Good morning, and thank you for the opportunity to present today. My name is John Ridge, and I'm presenting on behalf of Lumino DX regarding reconsideration of the CMS proposed crosswalk of PLA code 0575U to 0069U. Code 057U describes a transplantation medicine assay for liver allograft rejection that utilizes miRNA gene expression profiling by reverse transcription PCR of 4 gene targets: MIR 122, MIR 885, a housekeeping control, and a spike in control, using serum with algorithmic analysis, reporting a risk of a rejection. Today, I'd like to briefly review the clinical purpose of the assay, methodological and operational characteristics of 057U, address why 0069U doesn't seem to be an appropriate fit, and why 81595 represents a better fit. Next slide, please. Liver allograft rejection remains a major cause of shortened graft survival and treatment-related morbidity. First-year rejection rates remain significant, and current patient monitoring often relies on clinical assessment, traditional laboratory testing, and invasive tissue biopsies. HepatoTrack miRNA profiling was developed to provide a minimally invasive molecular approach to monitoring liver allograft rejection risk and assessing liver allograft status during immunosuppressive therapy management. Importantly, this is not a single analyte assay. This test involves circulating miRNA extraction, reverse transcription, real-time PCR analysis, housekeeping and spike-in controls, normalization workflows, and quality control review, along with, as I mentioned, algorithmic interpretation. The operational analytical workflow, therefore, is much more consistent with high-complexity molecular diagnostics than routine single analyte laboratory testing. Next slide, please. The current CMS crosswalk comparator 0096U differs substantially from 0575U in several dimensions that are material to CLFS pricing. First, the assays operate in completely different clinical settings. 0575U is a transplantation medicine assay used to monitor liver allograft rejection risk. 0069U is an oncology assay evaluating colorectal tumor biology. The specimen types are fundamentally different. 0575U uses serum-based circulating microRNA analysis. 069U utilizes formalin-fixed paraffin-embedded tumor tissue. These specimen differences drive substantial distinction in specimen handling, extraction workflows, quality control requirements, along with staffing needs and operational and resource utilization differences. Additionally, 0575U incorporates multiple analytes and multiple controls, whereas 0069U evaluates a single miRNA target. While both assays utilize RT-PCR methodologies, methodological overlap alone does not establish overall comparability under the CMS crosswalk framework. Next slide. We respectfully believe CPT 81595 represents a substantially stronger comparator to 0575U. Although the assays evaluate different transplanted organs and different RNA targets, mRNA versus miRNA, the overall molecular workflow, architecture, analytical sophistication, and intended clinical function are highly compatible. Both assays operate within the transplantation medicine. They both use blood-derived specimens. Both involve nucleic acid extraction, convert RNA into cDNA, utilize real-time PCR methodologies, incorporate normalization strategies and internal controls, and generate proprietary algorithmic rejection risk classifications. Importantly, while 8159 evaluates more total gene targets, 0575U induces additional complexity through miRNA extraction and detection challenges, low abundant circulating RNA handling, spike and extraction controls, RNA stabilization considerations, and highly controlled normalization workflows. As a result, the analytical sophistication, molecular processing requirements, quality management burden, and algorithmic interpretation demands of both assays are comparable despite differences in analyte type and target count. Next slide. This slide further illustrates the substantial methodological similarity between 81595 and 0575U. Both assays include, at a high level, specimen collection, specimen verification, nucleic acid extraction, reverse transcription, PCR amplification, quality control review, normalization workflows, algorithmic interpretation, and final physician-directed reporting. The primary distinction is that 0575U utilizes miRNA analysis with spike and extraction controls and an additional miRNA extension step during miRNA detection, which introduces additional complexity associated with low abundance circulating miRNA workflows. Operationally, both tests function as high complexity transplant rejection molecular diagnostics utilizing RNA expression profiling and proprietary algorithmic interpretation. In closing, we respectfully believe that 0069U does not adequately reflect clinical use, specimen requirements, workflow complexity, or operational resource utilization associated with 0575U. And blood-based transplant rejection molecular assays such as 8195 provide substantially stronger methodological and clinical comparability. We appreciate CMS's consideration of these comments and thank you for the opportunity to present today. Appreciate everybody's time.
Thank you. Precision Medicine Care. Are you available?
Yes. Hi again.
Okay. Hi, we have your slides. Karen, you can proceed with recording. You can keep it.
So let's start from the beginning, or I guess so. All right. Okay. Should I start? Thank you for the opportunity to present. My name is Sherry Dadgar. I am board certified clinical molecular geneticist. I'm serving as a technical lab director along with Dr. Ehsan Nobach, who is a board certified nephrologist. We're presenting the PLA code 0628U for RenaDX product, which is a comprehensive renal disease genetics panel. To provide background, chronic kidney disease, also known as CKD, affects approximately 37 million of Americans. Up to 20% of CKD cases may have an underlying genetic cause. Major nephrology guidelines, including KDIGO, recommend consideration of genetic testing in evaluation of chronic kidney disease. RenaDX is a comprehensive genetic test panel for CKD that utilizes sliced exome sequencing to identify diagnostic variant and risk alleles. It analyzes 449 clinically relevant and nephrology-related genes. Clinical impact of the panel includes accurate and definite molecular genetic diagnosis. It changes or refines the diagnosis in a significant proportion of patients. Alters clinical management. Examples include avoiding unnecessary immunosuppression, guiding targeted therapy, or adjusting transplant planning. It enables family screening, as well as family management by identifying at-risk relatives before disease progression or before family pregnancy. It ends the diagnosis odyssey and reduces the repeated biopsies and costly workups. Therefore, the result of RenaDX can drive appropriate clinical management of CKD as part of the patient evaluation of CKD etiology. Nephrologists and other healthcare providers can order genetic tests in the workup of CKD symptoms and signs. To review methodology, RenaDX is a high-complexity sliced exome sequencing panel comparable to existing large multi-gene NGS panels under CPT code 81455 with multiple additional substantial bioinformatics platform to cover difficult-to-interpret genomic regions as well as professional phenotypic-driven interpretive burden. Next slide, please. We are using Illumina sequencing platform along with the several other pipeline to perform instruments to perform the test. The process includes DNA extraction, library preparation, target enrichment, and sequencing. This follows by single workflow bioinformatics that are usually done according to ACMG and CAP interpretation guidelines. We're using Genex and Imagine to perform primary, secondary, and tertiary analysis. We then further conduct an additional bioinformatics analysis for difficult to interpret genomic regions, including repeated regions as well as copy number variants. SharkV and Typer is used for highly repeated regions, and Clean CNV is used for copy number variation. Additional analyses are beneficial due to enhancement of interpretation and diagnostic yield. The process is then followed by phenotypic-driven interpretation approach for large panel performed by expert scientists. And then that follows by orthogonal confirmation. Scientific reporting and review is then performed by board-certified clinical molecular geneticists, LGG geneticists, or molecular pathologists. Our turnaround time is usually between 10 to 21 business days. Next slide, please. We recommend crosswalk to CPT code 81455, which is used for NGS panel of more than 50 genes that has single workflow as compared to RenaDX with additional analysis burden. The average reimbursement for the code 81455 is $2,850. We request a reimbursement amount of $3,250 per test for RenaDX. Our actual cost for analysis in the facility for the RenaDX is $2,855, and the cost justification are listed below. Rationale behind this is a clear comparator exists. CPT code 81455 describes genetic NGS panels of more than 50 genes in a single analytical workflow, exactly what RenaDX performs with 449 nephrology genes. Both 81455 and RenaDX involve large panel NGS with integrated sequence variant and CMV analysis, expert interpretation, and a unified clinical report. I would like to finish emphasizing that genetic testing in kidney disease reduces diagnosis cost by 20 to 41% and leads to sustained management changes in 86% of positive cases. With annual Medicare dialysis costs exceeding $88,000 per patient, even modest delay in dialysis initiation yields significant savings. Thank you so much for your attention. I'll take any question that you may have. Thank you.
Hi, Sherry. This is Mark
Hi.
Rumpler with the panel. What's your typical batch size with this run? This
The
NGS
typical batch size, it depends on the the volume that we have. It can vary from 8 to 24, around that range.
Okay, thank you.
Of course. Yes, thank you. Go ahead. I guess someone else has a question.
Hello, this is Joe Lennox from the FACA panel. Can you hear me?
Yes.
So hi, Joe Leonard from the FACA panel. I have a question. Is this performed from peripheral blood, or what is the substrate, or are you using affected kidney tissue for this analysis?
A great question. The specimens that we validated in our lab, this is the hereditary genetic testing. To clarify, we validated our assay for blood specimen, saliva, as well as nail clips.
So this is, for all intents and purposes, a germline test,
That is
so to
correct.
speak. Okay,
Absolutely.
thank you very much for clarifying.
Of course.
Thank you.
Thank you. I appreciate it.
Galaxy Diagnostics.
Hello, my name is Hannah Mamushka. I am from Alva 10 on behalf of Galaxy Diagnostics, joined by Jennifer Miller, VP of Clinical and Scientific Operations for Galaxy on the phone. I'm here to discuss code X287U, a single multiplex digital PCR assay that detects DNA from 3 tick-borne pathogen genera, Bartonella, Borrelia, and Babesia, at the genus level from 1 whole blood specimen with a qualitative positive or negative call for each. Lyme disease is very much in the news. Antibody-based testing, including the standard 2-tier ELISA plus Western blot algorithm, detects host response rather than the pathogen directly, which gives poor sensitivity in early infection because antibodies have not yet been developed, and leaves it unable to distinguish active infection from past exposure, since IgG antibodies can persist for years after clearance. Tests for vector-borne pathogens must be both highly sensitive and multiplex. These 3 pathogens circulate at very low levels in blood. They frequently co-occur, and they present with overlapping nonspecific symptoms. That combination is what hinders most conventional workups. Sequential single pathogen testing delays diagnosis, and importantly, very frequently misses co-infection. These are stealth pathogens, so direct, highly sensitive detection is often the only way to reliably find them, and the chronic sequelae they cause are significant: organ damage, immune conditions, debilitating fatigue, post-exertional malaise, cognitive impairment, musculoskeletal pain, to name a few. Critically, each genus requires a different antimicrobial regimen for optimal disease resolution. Determining which pathogen or pathogens are present doesn't just correctly diagnose, it specifically informs therapy. Direct multiplex detection of all 3 is the most reliable way to confirm or rule out infection at the point of clinical decision. The work behind that result really drives this crosswalk. The architecture is 1 specimen, 1 multiplex reaction, 3 independent analytical lanes. A single EDTA whole blood draw, DNA extraction, and then multiplex digital PCR run on the QIAcuity platform across 26,000 partitions using 3 independent TACMAN probe sets. The important point for this committee is that multiplex here does not mean 1 test that happens to look at 3 things. It means 3 complete independent assays executed inside 1 reaction. Each genus has its own independently designed and validated primer probe chemistry, probe limiting TACMAN chemistry, each one separately validated for cross-reactivity against the others. And each provides genus level coverage, not a single strain. This covers 31 Bartonella species, 13 Borrelia species, and 24 Babesia species. This is broad genus coverage designed and validated 3 times over. The interpretation is equally important. 3 separate analytical algorithms apply per target thresholds against the 26,000 partition fluorescence data. Each genus carries its own positive and negative controls, its own per target acceptance criteria, and its own per target sign-off. There's no shared call. Each of the 3 results is generated, controlled, and released on its own. Then there's the quality program. There's no standardized analytical reference material and no proficiency testing that exists for multiplex molecular assays combining these three pathogens, none. Galaxy had to develop its own quality controls and PT specimens in-house for each target specifically for this assay. That is real additional per target development work that doesn't exist off the shelf. And this is not new methodology. Galaxy already operates 2 finalized PLA codes in this exact clinical area on this exact clinical platform. 0301U, Bartonella droplet digital PCR on whole blood. 0302U, Bartonella droplet digital PCR and enrichment fluid. X287U takes that same validated ddPCR methodology and extends it from 1 pathogen to 3 genera in a single reaction. When you total the resource picture, 3 independent chemistries, 3 validation efforts, 3 sets of thresholds, 3 QC and PT programs, 3 independent calls, the per target work scales essentially linearly, 3 genre, 3 full analytical pipelines. Out of scope for this discussion, but again, to illustrate how difficult it is to work with these pathogens and detect them, Bartonella is shed into the bloodstream in cyclical waves. To guard against false positive caused by a blood draw occurring in a time when there's little to no Bartonella circulating, Galaxy runs an entirely parallel procedure with a separate aliquot of blood in which they perform an enrichment on Bartonella and a separate digital PCR assay. That scaling linearly is the basis of our crosswalk recommendation, 0301U times 3, which is $788.16. The logic is direct. 0301U is the methodology matched anchor, same digital pathology PCR, same whole blood specimen, same Galaxy CLIA certified laboratory. 0301U is single pathogen at 262.72. X287U applies that identical methodology across 3 genera. And as I've walked through, the work scales per target. 3 times the anchor is 788.16. This is also established CMS practice. Multi-target multiplier crosswalks have been finalized across multiple recent cycles. 0464U is Cologuard Plus, which was crosswalked at 81,327 times 3. 0482U, Mayo's preeclampsia test at 0243U times 2. Same lab, PLA to PLA target multiplier, and most recently, 0251U in the last cycle at 86200 times 4, confirming the CMS continues to apply per target multipliers in the current cycle. And that practice goes back to 2019. So our recommendation is to crosswalk to 030U times 3 at 788.16. Methodology matched, per target, and consistent with finalized CMS precedent. We're happy to take questions.